Hello, welcome to STEM at Home with Newton's Road. We are so excited that you came here to join us today, and we have some wonderful guests here that you're just going to have a fantastic time. So, the next slide, please, Annalisa. And I should say, I'm Barb Tremont. I am the Executive Director of Newton's Road, and I have with me here Annalisa Wiesner, our MTU, Michigan Tech University intern. And we have several special guests today, all from S.T.E.M, Ecology and Education Centers. We have Brett Bontrager, who's been working in non-traditional education for over 15 years in the Kalkaska area and loves to have children outside and learning and having fun. We also have with us Joe Kreider, who works in the forest area middle schools with seeds and has worked with youth in residential treatment camps and school settings for over 15 years. And I think all seeds people have a passion for getting kids outdoors. And we also have Elizabeth here, also with us from Seeds. So before we get started in that area, this is a live show that's being recorded. You will be able to see the recording afterwards. And if you are participating in the Zoom Live, we have a few tips for you on how to use these features that Matt is going to share.
Hello, my name is Matthew Kern. I am the station manager at Traverse Area Community Media. You can see here on this slide, if you're watching on Facebook Live, you can join on Zoom and interact with us in a couple different ways. You can also watch this live on channel 189 on Thursdays at 2 p.m. every Thursday at 2 p.m. Through the end of May and into the second week of June. And if you join on the Zoom app, you can communicate with us by using the chat button at the bottom center of the screen or the Q&A button as well to ask questions. And if you'd like to ask a question live on the air, you can use the hand raise button and we will unmute your microphone so you can ask the question. Also, if you're on Facebook Live, you can put your questions in the comments to this video, and we'll do our best to ask those questions of our guests.
Fantastic, thank you. We also want to do a brief introduction to Newton's Road. We are a STEM learning and career exploration nonprofit in serving the 5-county region where you all live. And to give you a little bit of more information about us, we're going to share our website. You can see that if you want to get to any of these STEM at Home programs, we're doing 4 a week. You just have to click this alert at the top of our website and it'll take you to all of that information on the STEM at Home page. Back on that home page, if you just click on the home page instead of on that alert, you will scroll down and you can get a sign up for our mailing list, and you will get a monthly update from our newsletter about all the cool things that are going on with Newton's Road, but also with all the STEAM makers in our region like Seeds, who's joining us today. And also we have a calendar that has our events, and we also publicize from the STEAM makers and other STEM related programs in the area. So we have a live calendar, like you can see Annalisa is pointing out to the Traverse Area District Library has some live shows, and there's other people who, there's a Challenge Island there I can see on our live. You can also view just for anytime science and anytime STEM, and this means it's something that you can do anytime during the day. And if your kids did not get any science from their teacher for that day, or they just love that topic, that there's some family fun activities that you can do with common materials at home. And so there's lots more things there on your website. We hope you have a moment to explore. But right now, we want to get to our special guests. So I'm going to pass it over to Brett, I think.
Hello.
Hello, Brett.
Hello, how are you?
Great. We are so excited to have you guys with us here today. What are you going to teach us about?
Well, it's great to be here. We thought we'd try to do something that we normally do with our kids in the late spring with everybody, and we don't really have a chance to get together and all get inside of the woods together. So we went ahead of time and we went down to Rapid River, which is located just on a border of our school property. And we went into the water and collected what a lot of people like to call water bugs, but I would say water insects because bugs are a little bit different. And this is normally something we do with our children to kind of check out how our river and stream are doing health-wise. And if you want to switch to the PowerPoint, we'll start talking about how it works. So in the first slide, we call it biomonitoring, which is what a lot of people refer to it as. And what that really means is we are just checking out the biological health of the environment in a specific, with us it's a specific region or a specific watershed look. We're going to do it, you can do a lot of different monitoring, but we like to focus on the critters. You can do like chemical monitoring where you go down to check for pH level and different things that are within the water. But another way that you can do it that you can really check the health is just seeing what is able to live inside your water. And the organisms living in the stream are going to experience all of the conditions of that stream or river, the good and the bad, and that's going to determine who's hanging out and who's surviving in the stream. So one way that we can check the health is by going and seeing which organisms are living in that stream. Next slide.
And real quick, we do this at Forest Area too, and we don't have a river on site like Brett does, but we do it in our pond, so it can be different bodies of water. It doesn't necessarily have to be a river.
Yeah. Yep. Now there's easy ways to do this at home without all the equipment that we may use here. We can talk about that later. Why use macroinvertebrates for biomonitoring? Well, they're really good indicators of stream health because you're going to know right away whether or not a stream is healthy based on how many of the different species. You're looking for a biodiversity. You're looking for lots of different species of these little critters and certain amounts of numbers. And there are things that are always common or abundant with it, and you're looking for some of those, but there are also some species that are specifically very intolerant of nasty water, whether that be a chemical thing, whether that be, for whatever reason, a lot of sediment in the water. They just can't cut it. So some of those things will disappear, whether they're leaving or whether they're actually just dying off completely. Also with that, you can look at the individuals and you can kind of get the cumulative impact of the pollution in direct regions. The reason it's kind of easy is because if you pick a section that you can do every time, it fits with these species because they barely move beyond their region. Like they're very and when I mean region, I'm talking like hundreds of feet. They will live their whole life within hundreds of feet. So you kind of get this little micro, almost a microbiome, but not quite, for those creatures. And their health is going to reflect the habitat for that chunk of the stream or river. So it's something, if you want to do the whole river, there's of course different areas in the river, and you might do it throughout the whole river, but we just look at the area that's right next to our school. We can go to the next slide. So macroinvertebrates is a very large word, and some people might be thinking macro what? So macroinvertebrates are organisms that have a backbone. That's the vertebrate part. The macro part means that you can see it with your eyes. You don't need a microscope. You don't need to get a magnifying glass. These you can at least see with your eye. They might be really tiny, which some of the ones we'll look at are pretty tiny, but there are things that we can see and just kind of scoop out ourselves. There are lots of different microinvertebrates. You can get your bivalves, which are your clams and your mussels that you might find. You get your insects, nymphs, and larvae. So we're going to see some larvae. You can get some snails. But we're going to really focus on the insect nymphs and larvae with Rapid River, because we get some beetles, but we haven't really gotten any like crawfish or anything like that down there. And of course, everybody knows about the leeches that can be in there too, and leeches are considered microinvertebrates. So next slide. So what is a bioindicator? So a bioindicator is a living organism that gives researchers an idea of the health of an ecosystem. And you can observe microinvertebrates present at the site to understand the general health. And part of the thing to keep in mind, though, is that this is something that we should do over time to see relatively what is happening over time. If something were to, let's say there was some massive flooding and things got really messy out there in the spring, but it does like a self-clean basically. In a couple of years, we might be right back to normal, and we'll see that in our trends. Something could be happening upstream or downstream, and depending on where we're testing, that could happen. But so we're looking over time. If we were to see like species just disappear completely, like from one spring to the next, or from when we did it, let's say in the fall to the spring, we'd be a little worried. We'd want to check some stuff out.
And also with the seasonal, like what time of the year that you're doing it, you want to pay attention to that, right, Brett? Because like we didn't get any mayfly.
We got some there,
just missed
small.
Hatch. Yeah, not as many as we might.
We just had a big mayfly hatch over here a little while ago. So the size of the species that we're collecting, it's kind of the start of a new cycle. Let's just talk a little bit about how we categorize microinvertebrates. Next slide. When we're looking at testing to check the health of a stream, we're going to categorize them into their sensitivity to pollution. And of course, there's lots of different ways to sort them out, and we'll show you that later with the biological sort out. But what we're doing when we're testing is we're checking for the different sensitivity levels of these organisms. So some are very pollution sensitive, and if you were to have any kind of anything that would rear its head to dirty up your stream, or there's some logging and silt and things, it's just not clear anymore. There are some, or things are getting dumped in the river. These types of things they're going to disappear pretty fast. They're either going to move along, they're going to find a way to move along, or they're just going to die out. Mayflies are a good example of that around here. Water pennies, we get a lot of water pennies and mayflies. Those are very pollution sensitive. Stoneflies, which are not on our screen, but stoneflies are very pollution sensitive too. So we look for those. Moderately pollution sensitive, that's where you get into your hardier things like your crayfish, your dragonflies, and your clams. And then pollution tolerant species are the things that just are always everywhere. Black flies do what they want, leeches do what they want, and it doesn't really matter to them. Next. Getting into functional feeding groups, just what are these little guys eating? We always think about these little guys as getting eaten because that's what our trout and different fish like to eat when they're in the river. But they eat stuff too, and we can categorize them into what they eat. You have scrapers, water pennies, snails. Some types of mayflies, they'll scrape things off of the rocks and off of the bottom, and that's what they're chowing on. You have shredders, and they will go through different organic matter that falls into the water. Next slide. And then you have some collector gatherers, and they will also collect the organic matter from the bottom of the stream. And then you get the filterers who actually like filter things through themselves. And they'll filter the things that they like from the water. Next one. And lastly, you get into some of the more interesting ones for me anyway, and you get into the predators, and you get into the stoneflies, dragonflies, different things that feed on other consumers. One thing is we like to show the kids, we didn't get any big dragonfly larvae, so we have this video. If we could watch this video, and it'll tell you a little bit about the dragonfly life cycle, but also how the dragonfly, why it's such a great predator. Something about it that's kind of fun to watch. So we can watch that video.
is brought to you by Curiosity.
Can y'all hear it?
It needs to be a more higher volume.
Okay.
For over 300 million years, these lethal hunters have ruled the skies. They're the order Odonata. That's dragonflies and damselflies to most of us. Before the dinosaurs even existed, they had a 2-foot wingspan, like a small hawk. Today, they're more modest in scale, but no less deadly. Take their eyes. Each tiny hexagonal cell picks up light from a different direction, which gives dragonflies an almost 360-degree range of vision. 4 wings help them hover or turn on a dime. That means this hunter rarely misses. The weird thing is, Odonata spend most of their lives in a place where these killer piloting skills don't help. This is where their mothers lay their eggs. When they hatch, the babies, called larvae or nymphs, spend months or years underwater. Their wings are still growing, so they aren't any help in scoring a meal, like this tasty mosquito larva. It's a larva-eat-larva world down here. Did you see that? Let's slow it down. The nymph has a killer lip called a labium. Remind you of this creepy thing? For this skimmer nymph, it's shaped like a spork. Only dragonfly and damselfly nymphs have this special lip. This kind of dragonfly nymph, a darner, has an extra surprise. There's a pair of pincers right at the end. It all happens in a fraction of a second. Think of the lip as a knife, fork, and plate all rolled into one. When the meal is over, it folds up neatly, ready for the next occasion. These baby skeeters don't stand a chance. And that's good for us. Let's hope it stays this way for a few million more years.
This episode is brought to you by CuriosityStream, a subscription streaming service that offers documentaries and nonfiction titles from some of the world's best filmmakers, including exclusive originals. Want to learn more about dragonflies and other insects? CuriosityStream's video series, Insect Dissection, investigates how insects...
With the dragonfly nymphs, they're pretty cool. They have different ways to get their prey than other predatory insects in the water. And I just wanted to show that. It showed a pretty good example of the life cycle, how it all works, the eggs to the larvae to the full nymphs. And they can spend quite a bit of time in the water as nymphs and get very little time in the air. But I don't know, I just think those jaws are pretty cool. And if you get a big enough specimen, you can actually, if you're very careful, you can get it and you can draw out its jaw and you can show normally. We didn't find any big enough. We have some small ones, but it's not going to work. But so yeah, so the macroinvertebrates, there's a whole bunch of different kinds. Now your question is, what are around here? That's just going to kind of depend on your body of water or your stream or your river. What we do with the kids is we normally go out into the woods, and there's a number of us that get in the water. We have these handy nets. The nets that we use here, yeah. So they're kind of like filter nets. You kick up the stream, and then the stream will push everything into your net. And you pull it out, and you check to see what you got. And then you bring it over with what you have. You put them in, we use these, just these giant cake pans, basically. And we get a bunch of different specimens. We put them inside there. We figure out what we have, and when we think we have enough for a certain amount of time so that we're consistent through all of our recording, we bring them in. And then once we get them in here, we have to identify. If you would show the dichotomous key. Yeah. That key PDF. Yeah, so we got to look at something like this because you never know what you're going to get out there. Today, we got a pretty good assortment of stuff that you would see on the bottom row of that key right there. And we brought some of them in. We're going to throw them underneath the camera. We have a zoom-in camera that we're going to switch to, and we're going to go through how we look at them and how we look at this key to figure out what we have. And so what we would do normally is we would have multiple pans of these from a set amount of time. And we would begin to record how many of each species, after we've identified things, how many of each species we have, what species we have. And then we can start assigning points to how many of each species. So mayflies and stoneflies, for example, are they're like we talked about, they're very pollution intolerant. They don't want to be around that at all. So those are worth a little bit more on our scale because we know if we have those, things are looking pretty good for the health of our stream. If we're not seeing a lot of those, if we're seeing more in the middle, so all of them get graded, and then the kids do some math with figuring out multiplication. It's fun stuff. They get to give their stream a health rating. So right now, I'd like to show you some of the stuff that we pulled out just like an hour and a half ago.
Do you guys want to show that video here?
Oh, yeah, we have a video that's collecting.
Oh, I see. You're going to the other camera. That's fine. I'll stop sharing. Cool.
Yeah, let's do a little bit of our video collection, and then we'll switch back to the live bugs. Insects.
So you want me to show the video right now?
Yeah, if you would.
Okay, great. Okay.
All right, so we leave the school, we have our pans, we have our nets, and we're making our way down to the stream. We're very lucky to have a really nice river right next to our school. We're going through the cedars here, getting closer. Like we found some flowers. All right, so this is the one part that I would like to show a little more of. This is right there was a fast flowing section of the river. And then to the side there, what is in front of me is a slower like stream that goes into the river. Because of the different, the velocity of the water, you're going to find different things in each of the different flowing areas, along with what's on the bottom of the river in the stream. So there we're getting some water into the pans so that we're not putting our little guys into nothing. They just need their water. So yeah, the sandy area there. I'm gonna take some sandy samples first. And I'm gonna put the net down. At some point. Then put my foot in front of it. I'm going to start kicking it around, stirring up different creatures, different things that are underneath there. Then I'm going to pop it up and I'm going to look through it. And that's actually enough of that video right now. That's just I did that about 10 times, 5 in the sandy area, 5 in the rockier area. And let me show you a little bit of what we found. So we found some different critters out there. The first one I'd like to show you that I think is pretty cool is the caddisfly. And the caddisfly larvae looks like, let me see if I can get it under there. Oop, wrong way. He's right there. So what that is around him, caddisflies like to build themselves little homes that they stay in. until they outgrow them. And then they leave that home and they go create another one. This type of caddisfly, and there are different types of caddisflies that use different materials. That is actually like leaf debris, plant debris. Some of them will actually use rocks and different pebbles when they get big enough, and they'll make like pebble homes. Some people will take these and they will put tiny jewels and different pieces of exotic metals, and they'll actually put the caddisflies into those containers, and caddisflies will make structures out of jewels and different metals. There are people that make different arts and jewelry from the hard work of the caddisfly. But as you can see, this one that's just like plant material. No rocks on this one. Sometimes in the year we find the rockier ones. The next thing I'd like to show you is a guy that looks kind of like a squid. And it's called a scud because it kind of scoots around. It's kind of quick. Hopefully he'll be not camera shy. Oh, he's just going in circles. So we'll get him as he does his rounds. And I didn't really know anything about these before I started doing this. I didn't realize we had anything that was very shrimp-like in our water. Let's see if we can get him. He's fast. But it's very shrimp-like. If he would slow down here. No, he's gonna go in circles. But he's very shrimp-like. And I find a lot of them, they're pretty tolerant of different types of pollution and different types of water. So they're pretty easy to find. That's a fairly large one. You might find them a little bit larger. But I like those. They're quick. You get them in your pan, they're zipping all over the place. Another one that we found was, we didn't figure it completely out yet. I think it's a black fly larvae, but he's hanging out there on some of that debris. And there we go. They're pretty active. You'll find some of these out there. And I believe that that's a black fly larvae, but I could be wrong. It could be a crane fly larvae. I'm not very good with my little guys there with the worm-like stuff. That was a fun one. Then we found a bunch of mayflies out there, even after the last mayfly hatch. Got some different ones, slightly different colors. This one, I can't tell. It almost looks like it's what's left. They molt a lot, so I believe that that's what's left from a larger one molting. That's his little molten body there. And you'll find a lot of different things when you're kicking things up, and just watch for them in your nets. Yeah, kids just like getting out and getting these, checking them out, using the key. The older ones like to do the math to find out if it's a healthy stream. They're always waiting to see who's doing the last equations to figure out where we rate. But they all like collecting them. You can find some small beetles out there occasionally. Some very small, some bigger. Some years, yeah, that one's moving too. Some years we've found some predaceous diving beetles out there. And the one year we found one with a little trout minnow in its mouth. We really enjoyed that.
So this is something where if they, like on the last 2 weeks, you saw those mayflies, like wherever you walked, especially near any water, and then suddenly they were like gone. And is that because they laid eggs or something and now they're gone?
Yes, yep.
Okay.
So they spend most of their life in water, and they have their day or two of flight, and they take care of the creation of eggs, and then we repeat the whole cycle. So some of those mayflies that you see there are some of the new cycle, and they'll get bigger. Mayflies are, I like mayflies when they're easier to look at, bigger of course, but they're more segmented. You can see stuff on them. We couldn't find any big ones because we just had that whole hatch. So, but we found some small ones. The circle of life is continuing.
Great. Well, I think we're going to have to wrap up the show. It was so fascinating. I didn't realize the time. Thank you so much.
You're welcome.
I really appreciated seeing the outdoors and seeing those critters up close and live ones that you just caught today. So thanks to both of you, Brett and Joe and Elizabeth, who helped set it up. And thank you for joining us at STEM at Home, all of you who are joined us through Facebook and other means. And we also want to say a thank you to our sponsors and supporters who make this kind of programming possible. And if there's something you really liked, let us know, like put a note in the chat. And then lastly, we're going to end with showing you some other upcoming shows. So today at 4:30, we have STEM at Home for 10th and 12th graders, where you will learn about some programs at Northwestern Michigan College, NMC, in the technology areas. And then on Tuesday, we have 2 shows, the Parent Coffee Break from 2 to 2:45 with programming for your young ones so that you can have a break and meet with other parents or just have your young ones watch a cool science show. And then STEM at Home for 6th through 9th graders on Tuesday. So thank you so much, and we look forward to seeing you next time.
Thank you.
Thank you.